Okay, grade 12, so welcome back. We're going to continue on with photosynthesis, and this is going to complete the metabolism unit. So next week we're going to start my most favorite unit, which is all about systems. So we're going to go through the nervous system and through the endocrine system and probably the reproductive system. So that's more talking about physiology, so anybody interested in both kinetics or human kinetics as well as nursing or potentially becoming a doctor or a nurse practitioner or a midwife. This would be a really, really interesting unit, especially the nervous system. Usually people find really, really interesting understanding how the brain and nervous system works. Okay, but before we do that, we're just going to finish up on photosynthesis. This will be part 2 of it. So we're going to kind of go through the 2 parts of photosynthesis, which is right in the name, the photo part and then the synthesis part. Okay. All right, so let's go through this. Now, again, when we go through this, we're going to do it in a lot less detail than I would normally in class, just because I would normally take about a week to do this, but we're going to try and do it in a class. So we're going to skip out on some of the small details and kind of focus on the big overall picture. Okay, so this is the general idea. When we make photosynthesis, we need to make glucose. This is what we need to make. Okay? And we need to be able to make it from CO2 and water. And if you remember, whenever you take simple molecules like CO2 and water, and you want to make a complex molecule, you're going to need energy. Well, that energy is the solar energy, the sun's energy. The problem is it's not in the proper form. It's in radiant energy. So what we really need to do is we need to take this radiant energy and put it into ATP, which is a usable form. Then we can use that ATP to assemble CO2 and H2O together through that anabolic reaction to create glucose, that complex molecule. So that's what we essentially need to do. We need to take sun's energy, turn it into ATP so that we have enough energy to combine these molecules. Okay, I'm going to talk about this probably tomorrow. This is kind of just a kind of an overview for some of those students who are doing biology, chemistry, and physics, or even if you just did biology and chemistry, you have a really, really good understanding. If you understood some of the topics we went through, you should have a really good understanding now of kind of the natural world. And I'm just going to kind of touch upon that through the different energy conversions. But I'll leave that till tomorrow because it's really for some students that really have taken all 3. Okay, so photosynthesis is really, again, a 2-part process. We're going to focus on the first part right now, which is called the photo part. And it's called the light-dependent reactions. Now, that name kind of refers to how it should be called. So it depends on light-dependent reactions. So during this time period, the whole process is basically you got to trap the energy. Okay, you got to take radiant energy and turn it into ATP, which is our usable form, and then just a little bit of a precursor to ATP called NADPH, and that's really just kind of another very close form to ATP, but it's not quite ATP, but it's a kind of a precursor to ATP. Okay, so in the light-dependent reactions, it's part 1 of 2 of photosynthesis. This is the first part, the photo part. Now, it occurs in the chloroplast, okay, so you need to have chlorophyll, and then inside the chlorophyll is chloroplast, and it occurs at the thylakoid membrane. So basically all these little stacks, it's going to occur right on the membrane part of the stack. Okay? Remember we did plasma membranes? It's just on the membrane part of the stacks. And this is the whole purpose. The main purpose is to transform radiant energy, which is the sun's energy, into more usable forms, ATP and NADPH, which is just a loaded carrier about to become ATP. So that's the whole purpose of this first action. Now, during this first reaction, we call it a Z pattern. So we're going to keep referring to basically this diagram right here that I have. And why they call it a Z pattern is basically because the energy goes up, and then it goes back down, then it goes back up, and you can see it makes a Z on its side. So they often call it the Z pattern, but we're going to explain exactly kind of what's happening here. So this whole process here, which I'm going to kind of describe in detail, occurs in 4 steps. Okay? And this is the whole light-dependent reaction. Okay, so when we're all done this, we're going to have ATP. We're going to have NADPH and ATP. And that's what the result of the end of this process is we need to have some energy. Okay, then we're going to go on to the next step. So how do these 4 steps make energy? Okay. Well, the first step is photosystem 2. So that's what we kind of have down here. So that's just the name of it. It's called photosystem 2. Okay, and then the other one over here is called photosystem 1. You think they should have gone in order? That should be 1 and that should be 2. I agree with you. It's just I didn't pick the names. So this is what originally happens. Photosystem 2 absorbs a photon of light energy. Okay, if you know a lot about photons, P680 is a certain specific photon, a frequency of a photon. Okay? So basically, light comes down, and all light is a bunch of photons. Photons hits this photocenter too, and basically kicks an electron off water. So water's in the vicinity, and then as sunlight comes down, it basically kicks the electron off of water. And that's why plants need to have water, is to basically take that electron. So we're going to follow the electron. So the electron got excited from the photon, okay, and then the electron comes off water, and it's now considered an excited electron. It's got tons of energy. Okay. That's step one. Okay. The excited electron leaves PS2, photosystem 2, and goes to an electron receptor. That's where we are right there. Okay. Now, step 1b. So we're already here. Step 1b. Photosystem 2 P680, which we just talked about, now needs another electron to excite a photon. So P680 plus is capable of ripping apart water and taking an electron. After this electron's up here, okay, then it's just going to basically rip another water apart to refill it. It's going to keep ripping water apart. Okay, and P680 is going to keep using sunlight's photons to rip water apart and just keep this little process that I'm outlining in red over and over again. Okay, when the water is ripped apart, the hydrogens remain in the thylakoid, and then the oxygen is released by the plant. So when we rip water apart, okay, oxygen just goes off. So the plant basically just breathes out oxygen. So oxygen comes out, and then what we're left with is just H pluses inside the membrane. Okay, so now we have some H pluses inside the membrane. Now let's go on to Q. So I'm going to go back and I'm going to highlight where we were. So we got sunlight, excited the electron was up here. Now, step 2. From the electron acceptor, the electrons are transferred through a series of molecules called the electron transport chain. So the electron is going to go down this. And why it goes down is it's basically releasing energy. It got energy from the sunlight, and now it's going to release it. And how it's going to release it, it's going to release it by pumping H pluses into the thylakoid space. There was already H pluses there from when we split water, but now we're going to even pump more in there. This is kind of very similar to secondary active transport that we talked about. We're going to create a really, really high H plus area, okay, maybe really concentrated of H plus. Okay. Now, when they do this, this whole pumping is done by this called this B6F complex. Okay, we won't really go into it. That's a little bit more detail that we need, but that's what's pumping all the H pluses in. It's called the B6F complex. It's basically pumping all the H pluses into the thylakoid medium. So basically right down in here, we're going to have a whole bunch of H pluses. It's going to start getting very filled. Okay. All right, so now the electron is right here. They just released all of its energy. Now we go to step 3. Step 3, that electron. Let me go back. Okay, it was here, got excited, released all of its energy. It's now down here. Well, now it needs energy again. It just released it all. So now it's going to get another photon of energy. And this time it's a slightly different photon, different frequency, P700. Excites the electron becomes excited again. Okay, so now it's the same electron got excited, lost its energy, and now got it excited again. Okay. So now we've re-energized that electron. Go to the last stage. Okay, so the electron went here, up here, pumped all those H's out, got recharged. Now we're at the final stage. It's going to do the same kind of thing. It's going to go down the electron transport chain, okay, but it's going to create some NADPH at the end, which is really just a precursor. So now it's transferred that energy from sunlight into NADPH, which is kind of a precursor to ATP. Right, now we're not done yet. Remember that we had all these H pluses down there. Well, what are we going to use them for? We obviously wanted to pump a whole bunch of H pluses. What are we going to use them for? We're actually going to use them to make ATP, right? The actual usable form. So we're going to have NADPH made, but we're also going to have ATP made. And that's done through, it's not really a step, but we'll call it step 5. Basically, you built up all these H pluses, okay, in the thylakoid space, and now as those H pluses want to rush out because they're going along their concentration gradient, it makes a whole bunch of ATP from ADP. Okay. So the H gradient created is now used to create ATP from ADP. Okay, the hydrogens cross the thylakoid membrane through the stroma and then through this thing called ATP synthase. Okay, that was what was interrupted when the person got intoxicated with, I'm trying to think of it, cyanide. Okay. So this converts ADP to ATP through a process called phosphorylation. So in other words, it's going to create some ATP. So at the end of the light cycle, so we're at the end of cycle 1, the photo part, we end with ATP and NADPH. That's it. We don't have glucose yet. This is what we're ending with. So now we're going to go on to step 2 of photosynthesis, which is going to actually create the glucose with the ADP and ADPH that we made during step 1. So step 2 is the light-independent reactions, which does not require sunlight like step 1 does. It can do it whether sunlight's there or not, but it doesn't require it. Okay, so now let's go into step 2 of photosynthesis. Okay. We call this the light-independent reaction. And why we call it the light-independent reaction is it can occur inside the light or without the light. It doesn't need light. Okay, you've already got that energy from the photo part, step 1. So now this process, which we call the Calvin cycle, it's another way of calling it, can occur at nighttime or can occur during the day. Now, this is the Calvin cycle, and if we were in class, we would go through this much more in depth. So as I do go through this, it's going to be a little bit confusing. So again, you just kind of want the big ideas. Don't worry too much about the small ideas. But the whole purpose of the Calvin cycle is we're going to take CO2, okay, and we're going to take water, and we're going to end with some glucose molecules in the end. Okay, and this is basically the cycle. So recall that photosynthesis is a 2-part process. We just went through the photo part. Now we're going through the synthesis part. Okay, so the first part is often called the light-dependent, as it has already been covered. In simplistic terms, the light-dependent, part 1, was used to convert photon or light energy into NADPH and ATP. Now these 2 products are then going to be used for part 2, which we're going to be talking about, the synthesis, or synthesizing glucose. So the light-independent cycle, okay, it used to be called the dark cycle, but they changed that because it can happen at light time when light is out or when it's dark. So it's not just when it's dark. Is responsible for using ATP and NADPH from the light-dependent reactions to convert CO2 into glucose. Can occur in the presence or absence of light, which is why it's not called the dark cycle anymore. And the basic idea is you're going to take inorganic carbon. Okay, so when we call organic carbon, means it's come from living things, but CO2 doesn't. So we're going to take inorganic carbon and convert it into an organic carbon called G3P. And G3P is just a precursor to glucose. Okay, G3P can create glucose, can create fructose, and can create all kinds of different sugars. But it's really just the starting block of a sugar molecule. Now this process where we convert CO2 into glucose is called CO2 assimilation. Okay? And it's kind of broken down into 3 phases. We got to fix the CO2, we got to reduce it, and then we got to regenerate RUBP. Now, all of these are going to take place in the stroma of the chloroplast, not in the membrane. I'll kind of show you a picture of where that is. So it's going to be happening in here, in this gray area. Okay? Part 1, I'm going to change this to yellow. Part 1 was happening on these membranes right here that I'm highlighting. Okay, right on the membranes in here. And once I've created the energy, they send the energy out to the gray area, and part 2 will be happening in the gray area. Okay, so let's go through this. The first thing we need to do, if we're going to turn CO2 into glucose, is it's called carbon fixing. And the first thing you're going to basically do is you're going to take carbon from CO2 and you're going to bond it to this molecule called RUBP. Okay, it's called Rubisco. It's a very, very important molecule, RUBP. It creates basically a really unstable 6-carbon molecule because it's 5 carbons. You add CO2, now it becomes 6 carbons, which immediately breaks down into 2 3-carbon molecules called PGA. So I know this gets a little confusing, so let's kind of write down here. We started with CO2. I'm going to add it to RUBP. That turns into a 6-carbon unstable molecule for a very short period of time. We don't even give it a name because it's such a short period of time. And then it immediately breaks apart into 2 smaller molecules called PGAs. Okay? So that's step 1. I basically took CO2, combined it to RUBP, and then those broke apart, and now I have 2 PGAs, which are each 3 carbons. Okay. All right, next step. So basically I've gone from here, started with CO2, and I went to these. Okay, and these are each PGAs. Okay, and I got 2 of them. So that's where I am right now in the cycle. So here we were PGAs. Then we got a bunch of energy from the ATP and the NADPH, and then we become high energy G3P. Now, this can go on and make glucose, and a small amount does, and that's how we make glucose. But most of the stuff, most of these G3Ps are going to keep going, and they're going to keep going because they have to refill basically this little area right here so the cycle can keep going. If we took all those G3Ps out and made glucose, then the cycle would stop because you wouldn't have anything left to turn G3P back into RUBP and then have the cycle repeat. So most of them just stay in the cycle and keep going, and only a small amount comes out each time to make glucose. The next step is reduction. The second phase of the light-independent cycle is the reduction, or adding electrons, okay, or the gain of electrons is a reduction. If you remember oxidation reduction reactions. So the PGAs created in stage 1 are now in a low energy state and just need to be converted into a high energy state. They need to be given energy. So to complete this, they use ATP and NADH to basically take that energy that's in ATP and NADH and now add it to PGA. So we're going to kind of store it in the PGA. So when that happens, PGA is converted into G3P. Okay, I know there's a ton of terminology. That's just how microbiology is. There's a ton of little terms that we have to try and memorize. So PGA again gets turned into G3P, and G3P is now able to go two routes. Most of the G3Ps will not leave and just go on in the Calvin cycle, never actually making glucose, but just going back and then going through the cycle again. But a small number will go off and actually make glucose. So I'm going to go back to this cycle and go back to where we are here. So to kind of clarify, we started with basically CO2. Then we added RUBP. It turned into PGAs. Okay. Then PGAs get a bunch of energy, and when they get a bunch of energy, they turn into G3Ps. And once you have G3Ps, some will make glucose. But like I mentioned, most are just going to continue on in the Calvin cycle into the last stage, which is stage 3, which is to regenerate RUBP. The third phase is designed to regenerate an important molecule used at the very beginning of the Calvin cycle, RUBP, because again, the very first step is RUBP combines with carbon, so we got to make sure that we keep refilling the RUBP. So regenerating this molecule allows the cycle to continue. Without generation, the cycle would run out of RUBP, and this would prevent stage 1 of the carbon fixation from happening. So RUBP is regenerated by combining some G3 molecules, and since you are making something more complex, you need a little bit of ATP in order to do that. Okay. So just at the numbers at the very end, if you want to make one glucose molecule, so just a single glucose molecule, you need 6 CO2 molecules, or 6 turns of the Calvin cycle. So a plant needs to take in 6 CO2 molecules in order to make a 1 glucose molecule, which is again C6, okay, H12O6. That's why it requires 6 CO2s, because you have 6 in the sugar. So 6 turns of the Calvin cycle creates 12 total G3Ps. Okay? And only 2 of the G3Ps combine to make a glucose. The remaining 10 are used to regenerate RUBP. So the G3Ps that the plant makes can make glucose, it can make cellulose, it can make starch, it can make sucrose, it can make all of those different things. And that's why instead of calling it glucose, we call it G3P, because it can make a number of things besides. I know this part was in particularly confusing. As we go through photosynthesis, and also to add to the confusion, it's the fact that this isn't the most interesting topic. But again, like I mentioned before, that's the challenge in science is, can I learn things that aren't the most interesting? Okay? So this is a lecture that you may have to watch a couple times, and you can see in the learning check as to how I'm going to address this difficulty in learning all these different terms and how difficult my explanation just was, because I realized it was very confusing. I'm not unaware of that, but that is how we present the material. And it's difficult, and the task is, well, how can I take this material and break it down so that it's simple? Okay. Thanks, guys. Glucose. And that's it.